Bogone bupirima reka ele morena kawena reka ema ratswarana morena orejoge leseding waar die wilgers welig spruit, doringboom sy skadu sprei, soos ons groei in kundigheid, mag U ons vlei. Sien ons hoe hier lei met die hand, laat U sien rus oor ons land. Strong streams united flow, Africa stands proud and tall. As we learn, we trust, we know, God is in control. Bless us, O Lord, guide us with grace. May Northwest be blessed always. Bless us, O Lord, sien ons hier. Ore Jehovah Tsemporena, sien ons, bless us, O Lord.
And we know that all things work together for good to them that love God, to them who are the called according to His purpose. Ek lees dit ook graag in Afrikaans. Ons kan nou van een saak seker wees. God gebruik elke ding wat in ons lewens gebeur om sy doel te bereik. Vir ons wat vir God liefhet, werk alles tot ons beswil uit. Isn't it just a wonderful opportunity that God gives us as ordinary people to live out His purpose? Something I often think about is that, do we really take care enough to use these opportunities in our lives to seek and live His will? Tweedens, dan die wonderlike belofte, vir ons wat vir God liefhet, sal alles ten goeie uitwerk. Soms gebeur dinge nie soos ons het beplan nie, en soms gebeur dit nie op die tyd wat ons dit graag wil hê nie. Maar hoe geruststellend is dit nie, dat ons weet dat die Here wat in beheer is, die beste weet en dit vir ons nou ons beste laat uitwerk. So is ons vandag saam met Sandra dankbaar, dat haar harde werk, haar loyaliteit en haar passie geseën word en beloon word. Sandra, mag jy ook hierdie geleentheid gebruik om die Here se naam te verheerlik en ook sy doel daarmee te bereik. Let us pray. Dear Lord, today we want to bring all the glory to your mighty name. We want to thank you for your kindness and favor that you pour upon us. Here, in besonder dankie dat u Sandra beloon met hierdie wonderlike geleentheid. Here, ons bid dat Sandra ook hierdie geleentheid sal gebruik om u doel te bereik. Seën haar, rus haar toe met alles wat nodig is om suksesvol te wees in hierdie nuwe posisie en ook dit wat vir u vir haar daarmee beplan het. Ons loof en prys u naam. Amen.
Good evening, ladies and gentlemen. My name is Prof van der Westhuizen, and it's my great honor to recognize and welcome the following distinguished guests and functionaries in person and online here today. First of all, the guest tonight, Prof. Sandra Barnard. It's a special day for you, and you're obviously very welcome here tonight. Then Prof. Robert Balfour, the DVC for Teaching and Learning, who's the main functionary tonight. A special welcome to you. Then Professor Helen Drummond, the Deputy Dean for Teaching and Learning in the Faculty of Natural and Agricultural Sciences. Welcome to you. Then other members of the faculty management here tonight. Prof. Sarina Claessens, who's the Director for the School of Biological Sciences. Prof. Carlos Besuidenhout, the Research Director for UESM. Professor Christo Venter, I think it's over there. Prof. Christopher, Director of the Center for Space Research, and then several of our colleagues here tonight, a part of the department that Sandra belongs to. A special welcome to you in the procession here tonight. Then there's several colleagues and students that's probably listening here tonight and in attendance here from this and other faculties. A special welcome to you as colleagues of Sandra. So, ladies and gentlemen, as you know, an occasion like tonight is a very significant personal occasion for Sandra and her family, and also her close colleagues and friends. You would recognize that being recognized as a professor, or Afrikaans, a hoogleraar, after decades of, I would like to say suffering also, work suffering, bags of salt consumed, consistent achievements that she had to go through to achieve this recognition, is a path that those around her would have recognized with a different perspective. Her family would have seen it in a way, her colleagues would have recognized it in another way. But we are all very happy in equal measure for you tonight in this occasion. Wat dit betref, wil ek graag in die besonder vanaand jou man Skalk verwelkom. Jou ma Kay en jou skoonouers Skalk en Hester en jou suster Marna wat hier is. Kan ek net sien of waar jou naaste familie is, hulle almal hier. Baie welkom aan u. Dis 'n spesiale geleentheid. Het gebeur nie baie in 'n familie dat iemand 'n hoogleraar word in 'n familie nie. So ook vele vriende en familie van Sandra en haar familie wat van ver af gereis het, spesiale woord van welkom aan julle. With this, I'm going to hand over to Professor Drummond for the introduction. Thank you.
Thank you, Francois, for the words of welcome. My pleasant duty this evening is to introduce Professor Sandra to you. And I think as I do that, I would like to have her here so we could all see her, if you don't mind coming up. Okay, now Sandra Barnard was born Sandra du Plessis. She's the eldest of 3 children, and she completed her BSc degree at the University of the Free State in 1986, cum laude. Upon completion of her master's degree in plant physiology, also cum laude at the University of the Free State in 1992, she accepted a position as a chief research assistant in botany under the leadership of Professor van Staden at the University of KwaZulu-Natal in Pietermaritzburg. Her responsibilities were to manage the molecular biology research laboratory and to act as chief researcher of several research projects. She soon realized that her key strengths were research and teaching. And to further her career in this direction, she enrolled for a PhD part-time in 1993 at the University of KwaZulu-Natal in Pietermaritzburg. And she joined the Department of Botany at the Noordwes-Universiteit in 1996 as a full-time lecturer. She started her professional teaching career teaching both day scholars and distance learning UNISA students. During this time, she completed her PhD degree in plant molecular biology and graduated in 1998. As part of her research development at the NWU, she has acted as supervisor for 11 MSc and 6 PhD students, as well as 2 postdoctoral fellows. She served as co-supervisor for another 5 MSc and 1 PhD students. During her research career, Sandra has authored or co-authored 33 papers in accredited journals, 7 papers in proceedings of conferences, as well as 3 Water Research Commission reports. Sandra became a C-rated National Research Foundation scientist in 2015, and she currently holds a C2 NRF rating. Sandra acts as a reviewer for national and international accredited journals. She currently serves as a sub-editor on the editorial board of the South African Journal of Botany and acted on the steering committees of several WRC projects. Sandra is married to Skelk Barnard, and together they enjoy farm life and the outdoors. Good for a botanist. They enjoy traveling and visiting South Africa's many national parks, and she is also an avid horse lover and is keen on equestrian endurance riding. Right, I am going to give you your gown now, Sandra, if we can just stand here. You're going to have to put it right. Okay, ladies and gentlemen, I present to you Professor Sandra Barnard, and we look forward greatly to your talk, and good luck. Thank you.
Good evening, everyone, and also everybody joining us online. It is my privilege to talk here tonight, and I would like you to remember that I did not do this work alone, and I was assisted by my students, whom I'm very proud of, and that gave me this legacy. And then, of course, all my colleagues and my co-workers that had to either work with me or listen to me. This is all dedicated to you. Sorry, it's not moving on. Okay, there we go. I would just like to start off with a brief summary. By nature, I'm a scientist, and I've always been interested in living cells and the processes within those living cells. My special interest lies in water and the photosynthetic organisms that we found within. You may find me outside sampling water or walking through the veld, but it is just as likely that you will find me in the laboratory doing research in the lab and using DNA technologies. I use this DNA technologies to study the interaction between the environment and the plants that live therein. As my mentors, my colleagues, my collaborators, and my friends, I'm proud to call you all my peers. I would like to start off, and I'm going to concentrate on my key or my core focus research, and that is my water research. So all the guys that work with me on the DNA work, we will get to that hopefully in one more occasion. So the core focus of my research is harmful algal blooms and the associated water quality of South African freshwater resources. And I've built this research on 4 pillars, which are freshwater quality, phytoplankton assemblages or algal assemblages, phytoplankton ecophysiology, and then also molecular ecology. The research has developed, and my current aim is to investigate the effects of climate change, which is the result of extended droughts and floods, on water quality, the algal growth, and the aquatic ecosystems. And I would like to address this national crisis through quality research that is also internationally acceptable. Right, as you can see here, there are some photographs of the algae. Those are the things that we work off. The most notorious one is, of course, microcystis. And you can see the results as well at the different places that we have worked at. Freshwater reservoirs are the major drinking water resource, and together with the rivers, the fresh water in rivers, for us as South Africans, and also for our general uses such as agricultural, industrial, and also recreational activities. Algae and cyanobacteria occur naturally in these waters. However, certain species are known to form harmful algal blooms, which can cause extensive problems in the drinking water treatment industry. Cyanobacteria, in particular microcystis is well known for that, are widely responsible for many treatment problems. They are able to clog up filters, they produce toxins, and then they can also produce other organic compounds such as tastes and odors, which give to us as the consumer the effect that we are not very keen on using it or mistrusting it. The first research pillar that I would like to discuss is harmful algal bloom formation. In other words, when colonies of algae grow out of control, causing problems for water purification and the production of taste and odors as well as toxins. And here I would like to recognize my first PhD students, Dr. Michaela Kruskov, currently working for NIRAS, which is a consultancy in Finland. Dr. Karen Conradi, currently running the Envirotech laboratory in Wellington, and then my co-worker for many of these projects, Dr. Atharita Venter. Now, when we do this research, mass occurrences of cyanobacteria and green algae can occur in the enriched waters, especially of the Vaal River, as well as in several impoundments, and the most notorious of those impoundments are, of course, the Hartbeespoort Dam and the Roodeplaat Dam. And these bloom-forming bacteria, cyanobacteria, in particular the microcystis, can then produce a hepatotoxin. But what makes it difficult is that not all strains of these algal cells produce these toxins. And it's very difficult, you can't distinguish between them unless you use the DNA to distinguish between toxic cyanobacteria and non-toxic cyanobacteria. So this was the first thing that we started looking at, is which condition starts to trigger them to produce toxins, which conditions to trigger them to form harmful algal blooms, and why do they survive better than others? Well, the first thing that we found out is that toxin or toxic strains usually grow at higher temperatures. At 22 degrees or higher, will the microcystis form toxic blooms. What we also saw is that in the case, for example, in Oscillatoria, which is another cyanobacterium, what they do is they change themselves morphologically. In case of nutrient limitations, so if there's not enough nitrogen or phosphorus, they will break themselves up into smaller pieces and they will distribute themselves then in the water so that when new nutrients come in, they can take full advantage of the situation. In the case of some of the green algae, Chlamydomonas, that often also form blooms that can cause big problems for the water industry, we have seen that they have a special strategy, that they don't grow out of control when there's enough nutrients, but they can maintain a very high growth rate when there's no nutrients or very limited nutrients available. And we have seen that they can then form blooms as soon as the nutrients return to the water. All these results have meaning in fundamental sciences, and we have as such published various papers on it. But it also has an impact for all of us at home. What is that impact? Not only did we create or contribute to the basic science, but we also contribute to sustainable development. All these scenarios that we have tested can then act as indications for environmental conditions that can present a risk scenario for any drinking water treatment plant. Based on these scenarios, preventative measures can then be taken. And this may result in cost savings, and it will facilitate the production of safe drinking water all year around. So that is the first pillar. So we've seen that bacteria can cause a problem. So the next part was then, if we know in which situations they can cause problems and which will trigger them to form these blooms, can we forecast these blooms so that we can take action well ahead in time? And here I would like to recognize the work of Dr. Karen van Ginkel. She used to work with Department of Water and Sanitation, and later on she created her own consultancy business. Dr. Annelies Swanepoel, that is currently still working for Rand Water, running the analytical services. And then Prof. Friedrich Reknagel from the University of Adelaide. And also one of my M students, Simone Boyens. Karen came to me with the idea, and we used environmental modeling, and the ones that worked the best was hybrid evolutionary algorithms to predict algal-related impacts on drinking water purification. The managers of a drinking water treatment plant rely very heavily on the results of the monitoring and by the quality analysis of water before they decide on what to add to the water to make it safe and drinkable. However, these results may be delayed anything from 3 hours up to 14 days, and that is all based on factors such as sampling distance, the distance that the lab is away from them, the lab turnover, how many samples that lab has to handle, and specific methods that they use for these analyses. Therefore, it would be to the benefit of drinking water treatment plants, managers, and the production chemists to be able to forecast these future events and high cyanobacterial and alkyl cell concentrations in the source water. In our research, Dr. Karen van Ginkel tested 5 different types of models. She used 20 years of data of various very hypertrophic or very nutrient-enriched reservoirs or impoundments in the Gauteng area. And of the models that she tested, hybrid evolutionary algorithms were functioning the best with our data. This work was then taken further by Dr. Annelies Swanepoel as part of her PhD, and she used the water of the Vaal Dam, and she tested it for the largest drinking water treatment plant in the country, and that is Suikerbos. These results can then if you use this model, these results can then predict real time, 7 days, 14 days, and 21 days ahead. And we've developed these models for microcystis, anabaena, serratium, as well as toxin production. So we can forecast those mass occurrences of these algae, as well as the toxins that they produce, anything from 7 days up to 20 days ahead, very accurately. These hybrid evolutionary algorithms use physical data that is being used when they are monitoring the water, turbidity, temperature, secchi depth, but also chemical analysis, and then, of course, the biomass of the algae present in the water. The large Suikerbos drinking water treatment works have applied these predictive models, and they still do today, in the optimal functioning and as part of the weekly compliance report. Annelie has then developed in her research the safety plan for Suikerbos, and these models serve as part of the safety plan so that when you get alert level, they know what to do so that they can implement the safety precautions in time and also manage to save costs, because these things are not running constantly. Then the third pillar that I would like us to pay attention to is problems caused by these phytoplankton or algae in the drinking water treatment plants. So if you already have these treatment plants, the different processes that takes place to produce safe and clean drinking water. So what we did was we did many research projects that aid in the application of conventional water treatment techniques and the removal of phytoplankton cells and their secondary products, the toxins or the taste and odors. And here I would like to once again recognize my colleagues, Dr. Atherita Venter, Prof. Sanet Janse van Vieren, my students, Dr. Hendrik Everts from Rand Water, Dr. Charlene Janse van Rensburg from Midval Water Company. A key aspect of this research was investigating then these algae that come through different processes and how they manage to do that, and when they produce their secondary products, how the treatment plant can mitigate it. Now, the first example I would like to pay attention to is that of an algae that we call serratium. It is quite a big thing, and it can at times cause big problems in a drinking water treatment plant. It can block up the filters, it can produce secondary taste and odors, and it also meddles with the drinking water treatment process, in the sense that in the beginning they add what we call a flocculant, and all the dirt and things come together and form flocks that can then be easily removed. Now, this algae has figured this out and can change the surface charge on its outer cell wall so that it does not form a flock, and it uses its motility to swim out of the flock, break open the flocks, and then let all the cells through, and even sometimes into the final water. Now, what we have found is that you can actually use that surface charge to determine which coagulant and how strong the coagulant is that you can add to the water to remove or to make sure that that algae stay in a flock. We have also shown that pre-chlorination without causing cell lysis can be applied to render them immobile so that they can't swim out of the flocks and break up these flocks. The continuous increase of total chlorophyll, or that is just another way to say the amount of cells in our freshwater resources, is especially in the nutrient-enriched Middle Vaal River, is a source of primary challenge to treatment plants, especially in the case of Midvaal Water Company that we work with together a lot. Midvaal Water Company has been very brave and has managed to treat the water successfully for a variety of combinations of water treatment processes to comply with the national standards for drinking water. Midvaal gave us the opportunity to not only investigate the efficacy of pre-ozonation as a water treatment process, but they also let us investigate the effectiveness of wastewater recycling, so as to reduce costs, increase supply, but also at the same time not compromise final water quality. We have published many papers and we've worked very hard of that. I'm sure Charlene can relate to that, and Hendrik as well, and Sanet as well. And several recommendations could be made for the operational guidelines to improve the water purification in the event of a serratium bloom at Suikerbos treatment plant. The efficacy of the different pre-treatments such as pre-ozonation, pre-chlorination was determined, and the possibility of reusing water, because when you try to clean water, a lot of that water goes to waste. So to try and save on cost and make sure that we all have enough water, we try to reuse that water successfully. Challenges regarding the production of potable water from the Vaal River were identified more recently. Then I come to the last of the research pillars, and this is probably the one that gave us the most fun to do, and that is pillar 4, where we look at land use and the quality of freshwater resources and the algal diversity, and the development of nuisance algae. So what we do, how we live, how we keep ourselves busy has a direct impact when it rains or when we are at water on the water quality. And here again, I would like to acknowledge Dr. Venter, Prof. Sanet, Dr. Anatoly Levenets, Dr. Annelie Swanepoel, that was very brave to help us with this, Dr. Tienes Morgenthal, Mrs. Lucia Marais, Mrs. Henri Boshoff, and now also Mrs. Michaela Stolz. And again here, Simone, I don't know, I have her married name yet, but I know she got married, Mrs. Simone Boens. She was still Miss Simone Boens at that stage. And we all worked together, did a lot of field work, and most of our research started close to home, since the ecological health of the Mooi River system is very important for us to maintain the quality of the potable water for us in Potchefstroom and the surrounding areas. This system is under constant pressure from anthropogenic pollution arising from both agriculture and especially mining. Our research has shown that changes in land use can have a detrimental effect, such as the increase of salinity in the Mooi River from the Bovensteoog to where it converged with the Vaal River. But we have also seen that changes in mining can have a better effect on the water, and the water could improve, and we saw a big decrease in sulfates. We also looked at the Middle Vaal and the Kokemoespruit, which led to changes in the abiotic environmental pollution sources that obliges stakeholders to continuously monitor the water quality. In the case of the Kokemoespruit, research emphasized that the importance of a well-planned monitoring program together with selection of representing sample points to ensure integrated management practices. The evaluation and the review of the Kokemoespruit monitoring data had identified several shortcomings. For example, monitoring of recoverable cyanide and fecal coliform bacteria had to be replaced by dissolved cyanide and E. coli, respectively. We also did work on the Sabi, Sand and Marite rivers, and our studies on the impact of land use on water quality have shown that water quality is modified by activities and processes taking place both naturally and hydrologically, as well as due to the surrounding landscape. Increased nutrients, especially during periods of high rainfall, strong flows, have shown that nutrients will increase, E. coli, which is a bacteria, will increase, and found that it is indicative of the deteriorating water quality due to the increase in urbanization. So the results emphasized the effect of climate change as well, and the impact that land use have on the water quality, especially during climate change. It furthermore documented the dynamic algal assemblages in these systems. We worked on several rivers, like I've said, the Sabi River, the Sand, the Marite River, the Kokomoespruit, the Vaal River, or the Middle Vaal River, and then, of course, the Mooi River. We looked at the effect of deterioration of water quality on several reservoirs, such as the Klerkskraal Dam, the Boskop Dam, Potchefstroom Dam, Rietspruit Dam, as well as the Inyaka Dam. So what does the future hold for us? The problems that South Africans experience and face regarding water quality and quantity, eutrophication, pollution, climate change, water scarcity, and water demand allows for us in the water industry exciting water-related scientific research opportunities and case studies. But it also prompts a serious reflection on how we have managed to damage one of the very resources that we all rely on most, namely water. Each of us can contribute by being a citizen scientist, whether you help to teach the youth, like we are now trying in our new community engagement projects, or whether you just clean up your fishing spot when you leave, or just use water wisely at home. Every job counts. I view each of these research achievements as my legacy for the next generation. My students continuing on their chosen paths in the water industry is a triumph to me and to the future for all of us. Lastly, I would just like to thank the people that has made this research possible. For the funding, the National Research Foundation, the Water Research Commission, the Finnish Foreign Ministry. And then, of course, our partners in research, without whom we couldn't have done it. Rand Water has done so much for us in kind. Midvaal Water Company has provided us with so many opportunities for research. The Inkumati Usutu Catchment Management Agency, Sand Parks, and then also the Department of Agriculture, Fisheries and Forestry. Thank you very much. I hope you enjoy the rest of the evening with me.
Good evening, everyone. I'm Robert Balfour. I'm Deputy Vice Chancellor for Teaching and Learning, and on behalf of the university's management and also the faculty, we want to congratulate our Professor Sandra Barnard, on a really wonderful and very insightful talk this evening. I think it's always one of the key features of a professor is to make highly complex knowledge and highly detailed research accessible to a community, not all of whom are necessarily scholars in the field. And so for somebody like myself, who's a linguist, being able to gain insight, Prof. Sandra, into the work that you've done, the water research work in relation to algal blooms, and the capacity of these blooms to generate toxins that, of course, we would expect to poison the water, but then also the interesting insight that not all algal blooms release toxins into the system is really insightful, something that wasn't at least previously known to myself. So it's an opportunity in which we recognize several decades of work that have gone into your development, and of course the impact of your development on your students, on the people with whom you work, the scholars, the peers, and of course the students that you also teach. I think it's a wonderful thing that the research that you've done has had an impact on our communities, not only the local communities, but also very important systems in terms of water research around the country, and of course also in the water industry. The Midval reference and the work with that provider, I think, is also critical, so that we understand that the water that we drink every day is linked to the work that you've been doing, and we want to thank you for that contribution to the university, to scholarship, but also, I think, so importantly, to the community. So thank you, Sandra, for doing that with us. I want to also finally, in my note of recognition, to acknowledge the family and friends of Sandra who have supported her development over a period of time. And none of this would be possible, of course, without those loved ones and close people gathered here this evening. And it's really a pleasure to see you all as guests of the university tonight, together with the faculty. And of course, last but by no means least in my acknowledgement tonight is the faculty. The inauguration of a professor of this closeness to us, and a colleague with whom we've worked over a long period of time, Prof. Sandra, arises from an academic community that have gathered this evening to celebrate with you, to recognize with you your achievements, and of course to help us commit with you to a further few decades of wonderful research still to be done, students still to be supported and inspired, and communities that will still no doubt benefit from all that work you've done. So on behalf of the university, we have a certificate in recognition of your achievement that we would like to present this evening, and also a small token of our appreciation, Prof. Sandra, that I'll hand over to you now so that we can get a good photo op as well for us all. After that, we will have the closing this evening by Prof. Francois, and then we will rise to sing our national anthem, and at that point the academic procession after the national anthem will exit, and we will ask Prof. Sandra to be at the door. She will lead us out, and then everybody will have an opportunity to exit and congratulate her in person. So, Sandra, let me offer you our warmest and heartfelt appreciation. I think let's give Prof. Sandra a round of applause.
ladies and gentlemen, just to end the formalities, I would just quickly mention it was really interesting, Sandra, to hear your talk tonight, what you have been doing for us colleagues and myself, who has been 25 years a colleague of yours and never knowing really what you were doing. I recall a bit the nature of botanists with taking Anatoly on a road trip in the Kruger Park, and while we were watching the top predators on the one side of the car, and man-eating type predators, and keeping them busy, Anatoly was slipping out of the car, running with a glass bottle into the water, and collecting samples for algae research. So that was a bit of a feel of what excites the scholars of that type. So, colleagues, just in conclusion, just from the faculty side, we would like to thank Ashley. Ashley, where are you? She's probably busy somewhere. There they are. Yes, thanks, Ashley, for arranging this, along with Darlene. And finally, for Leslie Kok and Mariette van der Walt, always, who's recording this function. Thanks very much. And I think we're going to stand for the anthem now next, Ashley. Thank you.